The brain. It's arguably the most important organ, responsible for nearly everything you do. It's kind of like the king or queen leading the body. Now, a royal leader is always surrounded by knights or guards working to protect them. Your body has a similar defense for the brain, but instead of knights, it has a specialized set of cells called the blood-brain barrier. Now, this barrier is comprised of specialized endothelial cells that form the brain's capillaries and brain cells that physically wrap around them to provide another layer of protection. This barrier is vital to your health because it blocks toxins from entering the brain. So it's not surprising that its dysfunction is linked with a number of diseases, including multiple sclerosis, stroke, and the spread of cancer into the brain. However, a healthy barrier also poses challenge because it can block the delivery of medications that we want delivered to the brain. So given the tremendous motivation to understand the barrier, you'd think we'd have a firm grasp on how it works. But the reality is that this barrier is extremely hard to study. The endothelial cells tend to lose their unique properties once removed from the body. My project is to build a cell-based model that maintains the cell's natural function so we can study the barrier in the lab. I focus on replicating the brain's mechanical environment so the cells might act the way they do in the body based on what they physically feel. Typically, cells are grown on glass or plastic, but the brain is much softer than those surfaces. Think of taking a cell from the brain and growing it on this extremely hard glass surface. The cells can feel that difference in stiffness and might change in response. Think of a knight. They're probably going to protect their home castle or somewhere like it better than they would a castle in a completely different environment. So if our model is just like the brain, the cells might perform better than they would on a completely different substrate like glass. And that's exactly what we found. By comparing brain endothelial cells on glass with cells grown on gels that mimic different stiffnesses in the body, we found that cells on the brain-like gel improved barrier formation and increased presentation of a barrier-specific protein as evidenced using a green fluorescent biostain. These results suggest an improved barrier formation on the brain-like gel. And now we know to integrate a soft gel as one model parameter, and we can continue to build on our model until we fully replicate this vital barrier and use it to study its mechanisms involved in health and disease. Success of this project could mean less animal testing, new techniques to deliver treatments to the brain, and new therapeutics for diseases associated with the blood-brain barrier. Thank you.